Hi everyone, welcome back. So in this unit, we're going to talk a lot about genetics, and we have to start by discussing DNA, the molecule that contains all of our genetic information. Now the full name of DNA is deoxyribonucleic acid. This is a two-part molecule. There are two strands that make up DNA that form either side of the ladder that forms the DNA double helix structure. And these two strands are joined in the center by points of connection between individual nucleotides. And these nucleotides are ultimately the instruction book, the step-by-step building instructions to make all of the different proteins that an organism could possibly make. Now let's talk about the structure of DNA. Now, DNA is this long chain of linked molecules together, these nucleotides that are chained next to each other, one next to the other, next to the other. Now, this is another type of polymer, very much how a protein is a polymer of amino acids. These two chains are held together at the center, and they twist on each other, forming this double helix type structure. Now, the nucleotides that are linked up side to side are held together by a strong sugar phosphate backbone. That is the composition of the backbone. So we're going to see sugar molecules that are chained together with familiar phosphate groups. Now these DNA molecules are very, very long. They run thousands to hundreds of thousands to, in some cases, millions of nucleotides long. So they can contain considerable amounts of information. This is another view of that structure, and I really want to point to this backbone. I want us to understand all parts of this molecule, not just the information containing parts, like the actual nucleotide bases, but also the backbone that is made of familiar sugars, these deoxyribose sugars, which is where the name comes from, deoxyribonucleic acid, and then we have phosphate groups that link each of these sugars together. Now this sugar phosphate backbone, each of these sugars connects to one nucleotide. So the actual information of DNA is contained within those sequences of those A's, T's, C's, and G's: adenine, guanine, cytosine, and thymine. Opposite strands, this strand versus this strand, they are complementary to each other, meaning that they always match with their opposite nucleotide. There is a specific type of pairing that we see with nucleotides where A's will only pair with T's and G's will only pair with C's in DNA. This is what we call base pairing, complementary base pairing. Now, the order of A's, T's, C's, and G's on any of these molecules will communicate the building instructions for a protein. Essentially, which amino acids go first and next in the polymer of amino acids that becomes a protein. Now, that's not even all the information that's contained within DNA. That's just what a gene is, the instructions to make a specific protein. There's other information outside of genes that exist in the spaces between them on DNA. Now, if we look at the points of connection between these two strands of DNA, we've got the ladder shape that forms when two complementary nucleotides pair up. And where they pair up, they form hydrogen bonds that link them to each other. These hydrogen bonds are really weak. Independently, they're very easily broken and not super stable. But because there are so many of them, we have thousands, tens of thousands, even more of these nucleotides in a long chain, and each of them forms a connection with its complementary neighbor. Together, all of these hydrogen bonds become quite strong. Any one bond is easily broken, but all of the bonds together, very difficult. They're also very easy to form again. So this is the force that holds the two strands in a DNA molecule together. But if we look at the linkages between nucleotides on the same strand, we can see a much stronger covalent bond that forms. These types of bonds are much less fragile, much tougher and harder to break, that link sugars to phosphates, phosphates to sugars, and those sugars to those nucleotides. Now, a DNA molecule is very long, and each molecule is held to the opposite strand with millions of those hydrogen bonds. The molecule contains a lot of information. And that information is readable in one direction on one strand, and also different information is readable on the other strand, the opposite strand. An important thing to remember is that DNA is always read from one side to the other, from 5' to 3'. Now, what does that mean? 5' and 3' are just chemical terms that refer to like the positions of certain atoms in a molecule. I don't really need you to know what it means, but I really need you to know the order of DNA being read or copied. It always occurs from 5' to 3'. And importantly, these two strands that form the DNA molecule run anti-parallel to one another, meaning that one strand runs 5' to 3' on one side, and the other strand is flipped, so the orientation is opposite. Now, this molecule, because it's so doubled up like this, we have two different strands. They are complementary, but they are distinct and different from each other. It allows DNA to store genes on either strand of the molecule. Now, one strand is going to contain information for some genes, and the other strand is going to contain information for different genes. This is a way of stacking the information and organizing it in a more complex way. Each of these genes contains the instructions to make a specific protein that the cell really needs. If the organism possesses the gene, it means that it has the capacity to make that protein. Now, the totality of these genes, all of genetic information that is in this organism, is what we call the genome. The genome is something that's contained within the nucleoid region of the prokaryotes and the nuclei of eukaryotes. So depending on what organism we're talking about, it will be organized in a different way, placed in a different kind of structure. Now that genome might be broken apart into different chunks, large fragments that we call chromosomes. Now the number of chromosomes is going to vary a lot by species. Bacteria always have one big circular chromosome. I really want you to remember that bacterial genomes are circular, a single chromosome. Usually, they only have one copy of that chromosome, but that can vary. Some species can have multiple copies. Eukaryotes, though, may have a few or many chromosomes that are not circular, but they're linear. They are a single strand that has a start and an end. That's not the case for bacteria and archaea, because those ends, the start and the end position, come together and they fuse to form a circle. Now, eukaryotes can have quite a lot of chromosomes. For example, humans don't just have one chromosome, we have 23 distinct chromosomes. And then many eukaryotes can have lots of copies of those chromosomes. Some can have only one, but others can have two. Others can have 3, 4, 5, upwards of 12 and higher. Humans are diploid organisms, meaning that we have only 2 copies of our chromosomes. So instead of us having 23 total chromosomes in our cells, we actually have 46. 23 unique copies, but 2 copies of each. Now, in any cell, prokaryotic or eukaryotic, DNA is going to have to be stored. And when it's stored, it's not normally super condensed into chromosomes that we often see and think about when we look at like images of mitosis in action, where cells are actively dividing and pulling chromosomes apart from one another. Instead, DNA is loose. It is partially unfurled with that nucleotide ladder exposed. That's really important because DNA is only readable, the genes are only capable of being used to make proteins when the DNA is unraveled and exposed. If the DNA is condensed, those genes are silenced and prevented from being read, transcribed, and translated into proteins. This loose DNA, the stuff that is ready to be read and used, is what we call chromatin. It's still organized, though. I don't want to give the impression that it's totally disorganized here. It's no tangled mess. It's organized in specific ways. And prokaryotes and eukaryotes actually do this organization quite differently. So in a prokaryote, prokaryotes will organize their DNA in the nucleoid region, and actually it's partially pinned against the cell membrane, and that really helps keep it in some kind of order. And there are also other types of special organizational proteins that can take some genes and crunch them down, make them very condensed and hard to read, which means they are silenced and not readable, and others that allow the DNA to be unfurled and accessible, able to be read and used to make proteins. Eukaryotes, though, organize their DNA in the nucleus. So it's a separate organelle that is set aside, and its job exclusively is in the maintenance, protection, and general use of the DNA that is stored there. And our chromosomes, as eukaryotes, are wound around organizational proteins called histones. So it's a little bit different. Think about this kind of like how we used to wind up and organize wired earbuds. So if you just stick that stuff in your pocket and let it bounce around, it would turn into a big chaotic mess. But if you took those wired headphones and you wrapped them around your hand first, or you wrapped them around your phone, when you pulled them out of your pocket later, it was much more organized. Now, in addition to the main genome, bacteria may also possess plasmids. These are small, circular DNA fragments that contain only a few genes. Now, not all bacteria possess plasmids, but some do. Some possess one or two or many in some cases. And there can be a lot of interesting genes that occur on these plasmids. The genes that tend to be on plasmids versus those that are in the main genome tend to be related to different aspects of survival, like dealing with harsh environmental conditions, or sometimes producing certain toxins that allow them to survive better as pathogens, things like that. This is also where we tend to see a lot of antibiotic resistance genes. This is something that occurs in high frequency in bacteria, but it's also something that we see in some archaea and also some eukaryotes. There are some fungi, for example, that have plasmids. And these plasmids can replicate and are inherited independent of the regular genome. Now, when a cell divides, it inherits the whole genome and the plasmids that were on that cell as well. But these plasmids can also be passed around to other individuals through that horizontal gene transfer that we have explored in the past. Any kind of passage of genetic information that is not from ancestors to descendants, that is not from parent to children. Now, a lot of the plasmid's DNA is actually taken up by regulatory and replicating genes, things that allow the plasmid to actually make the proteins that it codes for and also perpetuate itself. And as I mentioned before, the encoded genes that are in these plasmids are really diverse. Some of them can be environmental resistance related, so dealing with, say, excessively hot conditions or excessively cold conditions. Super acidic or basic conditions, super salty conditions, so on and so on. Antibiotic resistance is also common. Toxin production and virulence factors for species that are very pathogenic. And also accessory metabolic pathways, things that allow the organism to metabolize different kinds of resources that it doesn't usually have the ability to. Those are things that are often found on a plasmid and often shared between bacteria. And bacteria can change their behavior dramatically depending on those plasmids. If an organism lacks a plasmid for certain virulence factors, it means they're not producing those. This can be the difference between a benign, non-disease causing bacterial species and a species that is incredibly destructive and virulent, a very effective pathogen. It can also mean the difference between a bacterium that is vulnerable to a certain antibiotic and one that is highly resistant.